Hello students, this is the last video of chapter Strategies for Enhancement in Food Production, and topic covered in this video is single cell protein and tissue culture. So let's start. Alternate source of protein for animal and human nutrition is called single cell protein. Or we can say that if we use microbes as a source of protein, it is called single cell protein. So if we use microbes as a source of protein, that is alternate source of protein for animal and human nutrition, it is called single cell protein. Example of single cell protein is spirulina. Spirulina is a blue-green algae or cyanobacteria. So it comes under kingdom Monera. Now let us look at advantages of single cell protein. Number one, this spirulina can be cultured on a large scale in waste material like animal manure, sewage, or even waste from potato processing unit. So we can grow spirulina even on waste materials like animal manure, sewage, and potato processing unit. Secondly, the spirulina is rich in protein, minerals, vitamins, carbohydrates, but low in fat. And last, its utilization also reduces environmental pollution. So, a question can be asked on single cell protein, which is spirulina, and here are its advantages. Now let us discuss about technique of tissue culture. In tissue culture, we isolate a plant part called explant and place it in a nutrient medium in a test tube. So in step 1, we have taken a plant part that is explant in a nutrient medium in a test tube. Explant is any part of the plant taken out and grown in a test tube. Now, in step number 2, this explant will differentiate into mass of undifferentiated cells that is called callus. So, this explant changes into callus in step number 2. In step number 3, this callus is transferred into a growth hormone, which is mainly cytokinin. So this callus is transferred in a growth hormone and in a test tube containing growth hormone. As a result, callus change into plantlet. And after that, in the last step, these plantlets are transferred into the soil. So this is the technique of tissue culture. Now, based on this technique, we can learn the definition of tissue culture. Tissue culture is defined as a technique by which whole new plant could be grown or could be regenerated from explant. So as you can see, we have grown the whole new plant from the explant. So this technique is called tissue culture. And the capacity of the plant to generate the whole new plant from explant is termed as totipotency. So this capacity of plant to generate whole new plant from explant is known as totipotency. Now, sometimes we get confused between tissue culture and micropropagation. Micropropagation means if we produce large number of plants through tissue culture, this is known as micropropagation. So, method of producing large number of plants through tissue culture is called micropropagation. Now let us look at advantages of tissue culture. In tissue culture, we produce plants that are genetically identical to themselves and also with the original plant from which they are grown. So these plants are known as clones because it is also a type of asexual reproduction. Secondly, by tissue culture, we can recover a healthy plant from a diseased plant. So how can we recover a healthy plant from a diseased plant by tissue culture? Because we can do so because the meristem is always free from virus even if the plant is infected. So even if a plant is infected with virus, but the meristem would be always free from virus. So if we remove the meristem from the diseased plant, we can grow a healthy plant from a diseased plant. If you have gained something from this video, please subscribe to my channel Ramji Chopra, like and comment.